This is the International AQA certificate for GCSE, and we're looking at photosynthesis today. We need to know the importance of plants in our everyday life and in the food chains, and as we know from our knowledge of food chains and ecosystems that plants are producers and they are found at the bottom of every food chain. They harness the sun's energy to generate their own food, therefore making their own proteins and biomass, and that biomass is then what is consumed by all organisms higher up the food chain, providing their energy. So without the plants at the bottom of the food chain, everything above it in every ecosystem would not be able to survive. They form the fuel for everything, as this is suggesting. There are certain criteria that we have to be aware of for photosynthesis. Photosynthesis itself means synthesis, making of food using light. The chemical reaction that takes place is driven by the energy from the sun in the form of that light, and it's captured in the leaves of the plants and converted through a series of reactions which make up the photosynthesis pathways to produce starch, which is a stored form of sugar, which they will then use for energy. Photo being light, synthesis putting it together and building or making. So, in a reaction, the summary of what's going on inside of a leaf. The leaf is the general structure. Inside of it, you should be familiar with the structure of a plant cell. Cell wall around the outside, made of cellulose, giving it strength and rigidity. Cell membrane just inside of that. Key features for photosynthesis, unique to a plant cell, are that it has chloroplasts here at the light capturing parts of the plant. These are the chloroplasts. So plants all need to have some various components for their reactions taken in. So through the roots, water is taken up, and we'll look at the transport of that in a later video, and carbon dioxide is taken in through the leaves. Please don't try and infer in any exam answers that the waterproof layer of the surface of a leaf is there to allow water to get in or block oxygen getting in. Water only goes in through the roots, CO2 only goes in through the leaves. You then need to be able to put that into some form of equation. And the classic equation you have to be able to describe in the word form and in the chemical form. So describing what it is to start with, we're saying that photosynthesis is that chemical reaction that takes place. It is in the chloroplast where this reaction occurs, not in the nucleus or anything like that, and it's to convert the carbon dioxide and water into glucose and oxygen. So to put that into a chemical reaction, a word equation of that chemical reaction is as follows. You need to be able to write that out. On the arrow, we have chlorophyll being the essential light capturing component, and light energy is what's driving these chemical reactions with a whole series of enzymes. So that's the same reaction taking place. So to put that into a chemical equation, we need to make sure we can write this out chemically and make sure that it's balanced. Carbon dioxide plus water, H2O, giving you glucose, and you need to know this formula for glucose, C6H12O6 plus oxygen in the form of O2. To balance it, we need to add 6's everywhere. 6O2, 6H2O, 6CO2. You must know these two equations off by heart. It's also worth knowing the internal structure of a leaf and being able to identify the key adaptations that enable it to carry out its functions specifically. So looking at a leaf, obviously this is looking at it side on, as if it's been chopped top you have the waxy cuticle. That gives a waterproof layer to enable any water that does hit the leaves to run down onto the roots where it can be taken up. Immediately underneath is the upper epidermis, below which is where the main photosynthetic cells are found. These long, rectangular, regularly shaped cells called palisade cells. Palisade mesophyll cells just means it's the layer, particular layer of cell, mesophyll. These are tightly packed, rectangular, so that they can fit next to each other, therefore giving them a nice large surface area for capturing more light, and that you can pack lots of them together to maximise light capture again. So you want as many cells as you can possibly get in that upper surface of the leaf. Immediately underneath that is another layer, another mesophyll layer, this time called the spongy mesophyll. Spongy because just like a sponge is full of air, making it squashy, this layer is also full of air spaces. These air spaces are going to be essential for the diffusion of gases, CO2 coming in, which will come in through the stoma, which is the hole in the leaf, pore, and diffuse through the air spaces to reach the cells that require the CO2 for photosynthesis to take place. And similarly, then your oxygen that's produced as a waste product can come out easily through the air spaces down through the stoma. At the bottom is another lower epidermis holding it all together, and sometimes there's a waxy cuticle, but less so than on the top. Surrounding the stoma, which is literally a hole, to control the opening and closing of that hole are guard cells. And these guard cells, there's a pair of them together, and when they swell up, they form this curved shape, meaning that the hole in the middle is this hole in the stoma through which the gases can diffuse through. And when these guard cells relax, and they lose their water, they are therefore closed, so the gap in the middle is now gone, therefore meaning that less CO2 and oxygen can get in. The main reason for that is not really to control CO2 and oxygen, but it's to control water loss. Water is obviously essential for photosynthesis. It's also a waste product of respiration, which all of these cells will be doing because they're alive. And the plant is regulating its water content by controlling water loss, opening or closing these stoma, depending on the condition in which it's found. We've already looked at the equations of photosynthesis. There's another summary of them. And we'll come back to factors affecting photosynthesis shortly. We need to understand what the uses of forming glucose are and some of the fate of starch. And here are some of the key uses of that glucose. Synthesis, so this is making, so they're going to use the starch in a stored form, or the glucose that is produced, to create various components that the plant requires for growth. So that might be various nitrogen-based products and proteins, just like we have our proteins to build our own muscles and tissues. Plants are doing the same as well. Storage, glucose is soluble, starch is insoluble, so the glucose is stored as is the plant's energy storage in starch grains. That's typically what you might find in potatoes and other such seeds and things like that that need a lot of energy to keep water. The glucose could be immediately used for respiration. Respiration happens in all living things, as we've already said, so the glucose was made, automatically used. That respiration provides the energy which is required for all chemical reactions and growth of living things. Also, the glucose molecules can be joined together to make a big, large molecule, which will be the cellulose. That's making more plant cell walls, again, to make more tissues and further the plant's growth. So lots of different uses of the glucose, and we can test the glucose in the leaves to prove that they are being formed as a result of photosynthesis and using light. And that's an experiment that you have carried out, and we can have a brief look at now as well. So iodine solution, ready brown to start with, turns blue-black in the presence of starch. And so therefore we can prove that photosynthesis takes place using light by carrying out one of the following experiments. So, this may be one that you've done, or maybe the one that they could give you in an exam question and ask you to identify some of the key features of it, of the experiment. And so to start with, you take a normal, living, healthy leaf, and it needs to be placed into the boiling water. It's going to be left there for 30 seconds. That's very important because not only is it going to melt the waxy layer over the surface, but it's going to soften the cell walls, allowing the ability for the iodine to get into the cells where the starch grains would be. From there, you would have removed the flame, because obviously the flame with ethanol, the alcohol, would be a very dangerous thing, so you have to make sure the flame is gone. And we pick up our now boiled leaf and put that into the warm ethanol, and that now extracts the chlorophyll. You can see the chlorophyll from inside the leaf has come out into the ethanol surrounding it. We can now take that leaf and rinse it off in some warm water again, and that will go on to allow more softening of the cell walls, allowing greater access for the iodine once again. We then remove the leaf, place it on a white tile, and we can start to add our iodine solution on the surface of it. And as you can see, where it doesn't touch the leaf, it is a reddy brown colour, and where the leaf had contained starch, it's gone to a blue-black colour. You could need to describe that experiment, and you may need to understand particular elements, explain why you carry out each step. So, a step on from that is to use different leaves from different conditions to explain why or prove that it is actually the light that has caused that starch to be produced. And a variegated leaf is one that is different colours. Rather than just being green all the way across, it's got different shades all over it, which is what's shown here. So you've got the dark green areas and the lighter, more yellow areas. These light yellow areas are where there are much less chlorophyll found in the leaf. And therefore, without the chlorophyll, there can be no photosynthesis taking place, meaning there'll be no starch being produced. So we're not only proving that light is required, we're going to prove that chlorophyll is required as well. And so, to react them together with the iodine, this is the pattern that you would see having used that experiment we've just looked at. Where the green, dark areas are with lots of chlorophyll, they went blue-black when tested with iodine, and where the pale areas without the chlorophyll, they remained the reddy-brown colour of the iodine. So, typically they might give you something like this, where they give you some leaves, they ask you of different conditions. If we add iodine onto each of them, we can see we get different patterns being produced. They could ask you to identify what might have happened from different conditions. So a leaf in normal conditions with light and carbon dioxide would probably be one that has got lots and lots of starch and therefore has gone blue-black. A leaf that's been covered by a black paper strip is going to have no starch where the black paper strip was covering because it blocked the light, therefore proving that light was required for photosynthesis, and starch present in the bits that were exposed to the light. If it's airtight, it's in plant C, then there's going to be no CO2, and the soda lime also is there to absorb any CO2 that might be found as well. So we would therefore find that there is no photosynthesis taking place because there's no CO2. If it's got sodium hydrogen carbonate in there, that might be providing oxygen, so again it produces lots and lots of starch again, and a variegated leaf would be one, as we've just looked at, showing that you get different coloured patches where the light and the chlorophyll are both able to work together. So that's a typical set of results that they might give you and expect you to be able to explain what has happened at each stage, or proving that light and CO2 and chlorophyll are all requirements for photosynthesis to take place. Finally, we need to consider the elements required for effective plant growth, and not only is photosynthesis going to be important with the provision of CO2 and water and chlorophyll, etc., we've already looked at, but also the other minerals and ions that are taken up through the plant's roots with the water at the same time. And coming up through the soil, there will be a huge array of different minerals and ions, and there's only a few key ones that you need to know about. So we've got to consider what we know about specialised cells and the structure of the root hair cells. So if this is your roots, if you analyse the root under a microscope, you'll see that it looks almost furry. It's furry because it's got these long projections, very fine hairs, called root hairs, sticking out of them. And these provide a much greater surface area through which diffusion and uptake of minerals and ions can actually take place all over the surface, rather than if this was square, just across one side of it, where there's a much smaller surface area. So those are crucial elements of the root hair cell itself. A thin membrane provides a shorter diffusion distance, therefore also increasing the rate of uptake by the roots. Crucial minerals that you need to know about are nitrates, taken up from the soil. These are essential in the formation of amino acids, which go on to form proteins as well. So therefore, without enough nitrates in the soil, there would be a very small plant produced. It would be stunted growth because it doesn't have enough to make proteins for its own growth. So shorter, smaller leaves, smaller plants, often yellow in appearance as well. Phosphates are required, usually in the form of phosphorus, and that's required for photosynthesis and respiration. The deficiency in phosphates would also mean small root formation, because again, it can't provide enough energy to grow the roots enough. And again, purple leaves is a characteristic thing for phosphate production. And potassium help the formation of enzymes, which are essential in photosynthesis. Again, if there's not enough photosynthesis, the leaves will start to turn yellow, and the protein formation and all of these means that there can be no chlorophyll production as well. So a slightly yellow appearance to those leaves too. So having understood some of the importance of key nitrates and other minerals and ions from the soil in plant growth, it's important to then go on to understand what happens for plants that grow in soils that are not rich in those elements. And this is often the case, or maybe on rainforest floors, or those plants that maybe live partway up trees, growing on the sides of other trees, or maybe even have aerial roots that stick out into the air. They are all going to struggle to obtain enough nitrates from the soil, which are, as we've said, crucial for the formation of amino acids, which will build the proteins to make their tissues. So, rather than relying on a deficient soil, some plants have evolved so that they can actually become carnivorous. Therefore, eating other live animals provides a secondary supplementary source of nitrates and protein, which they can effectively digest and use for their own benefit, in the same way that animals do. They eat ready-made food, digest it, and reuse it to build their own tissues. Examples of these carnivorous or even insectivorous, they're typically eating insect plants, might be the Venus flytrap, living in damp, boggy areas where the minerals might be washed away. So the insect crawls in, triggers the hairs in the trap, and allows it to close. Therefore, it can digest it and absorb the mineral, the nitrates from the insect. Similarly, here the pitcher plant has an attractive opening for the insects. The insect can then crawl inside, and down the bottom is a whole load of liquid which contains digestive enzymes, and that traps the animal, the insect inside of it. It will digest it and make use of it. And you can see this is actually suspended from another tree, so it's not attached to the ground for roots in the same way. So these are both examples of where plants have evolved to and adapted so that they can survive in low-level nitrate soils, which are